The kind of nature versus nurture debate, I think, is kind of sort of dead, for lack of a better term. The consensus is that it's both.
Welcome to High Five with Dr. Igor Grant, Distinguished Professor of Psychiatry and Director of the Center for Medicinal Cannabis Research at UC San Diego. Each episode will take you on a fascinating journey into the evolving world of medicinal cannabis, as Dr. Grant poses 5 thought-provoking questions to his guests, uncovering their personal journey and unique contributions to the field. Now, here's your host, Dr. Igor Grant.
Welcome back. My guest today is Alex Miller, Ph.D., Assistant Professor of Psychiatry at Indiana University School of Medicine. Dr. Miller completed his Ph.D. in clinical psychology at the University of Missouri and a postdoctoral fellowship in substance use disorder genetics at Washington University School of Medicine. His research focuses on applying genetics and neuroimaging data analysis to better understand the developmental causes of substance use disorders and impulsive personality traits as a risk factor for the onset and progression of substance use disorders. Welcome, Dr. Miller.
Hi, thanks for having me.
Appreciate your being here. So we're going to talk a bit about some of the exciting research that you've been doing. And just by way of introduction to our audience, there's a common idea that drug use changes young people's brains, maybe even leading to permanent brain injury. Another idea is that people who use drugs might have brain differences to begin with. And your work may begin to shed light on this chicken-egg problem, if I could call it that.
Yeah.
So, can you explain a little bit about how your research is conducted? I know that it's part of a larger study that is called the ABCD study. So maybe you could say a little bit about how your research is conducted and what is the ABCD study?
Sure. Yeah. So focusing on the ABCD study to start, the ABCD study is a nationwide study of almost 12,000 children recruited between 2016 and 2018 when they were between 9 and 11 years old. And these kids were recruited from 21 sites across the United States. So it's really kind of an unprecedented study in terms of its size and design, because they're following these kids over the next 10 years with mid-year, yearly, and assessments of a whole host of variables related to mental health and substance use and their environments and interactions with peers, things like that, as well as brain scans every 2 years. And they've also collected genetic data from these participants. And so it really provides an opportunity to kind of, on a very large scale, examine changes that occur across late childhood, early adolescence, into emerging adulthood, both in terms of mental health and substance use, but also the relationship of those things to changes in brain structure, brain function, how genetics plays a role, and how environment plays a role. And so this is a data set that is freely available to qualified researchers all across the world. So it's a well-studied, even at this point, data set. And so we were interested, like you mentioned, this sort of chicken and egg idea, and most of the prior literature on kind of the brain's relation to substance use and substance use disorders has kind of been conducted in smaller samples. So we're talking a few hundred, at the most, and they've largely been cross-sectional. So we're looking at people who already have a substance use disorder or use substances heavily and what their brains might look like compared to those who don't. And the ABC study kind of provides the opportunity to rewind the clock a little bit, right, and look at kids who, when the study started, most of them had never used substances at all. And so what we did was kind of look at that very first brain scan of the study, right, and looked at whether any differences during that initial brain scan were related to the likelihood of some of these kids going on to initiate substances. And when I say initiate substances at this stage, right, these are 9 to 15-year-old kids. We're talking about very small experimental kind of interactions with substances, but looking at whether brain differences that exist prior to these first earliest stages of substance use are related to that substance use.
Right. So, your work involves both genetics and neuroimaging, I know. Before we get into the neuroimaging, is there any evidence that, is genetics just baked into us, or can the environment and other factors also change how genetics express themselves?
Yeah, definitely. And that's kind of my primary area, right, is genetics. I'm a clinical psychologist by training, and kind of the tools I use to explore the etiology of substance use disorders are statistical genetics traditionally, and now more recently neuroimaging. And the kind of nature versus nurture debate, I think, is kind of sort of dead, for lack of a better term. I think most people the consensus is that it's both, right? That there are genetic influences that are inherited from our parents that can even interact with our environment, and potentially environment provided by our parents in ways that kind of provide either protection from different negative outcomes or make us kind of give us a higher risk for those types of outcomes.
Yeah. So, but I think what you're also finding, and others are finding, is that how the genes we're born with express themselves may vary from person to person, and even that expression may be conditioned on your life experiences, or even in this case, if you start using drugs, that can change how your genes are working and programming different vital life functions. Would that be right?
Yeah, I think that's the case. And I think there's also kind of some evidence, at least from twin studies, and this is sort of being explored more recently in kind of molecular genetic studies, that we're born with a certain genotype, right? Our DNA doesn't change a whole lot, but its relationship to different phenotypes can change based on the environments we're in, and it can change over time. So some of the genetic factors that promote certain traits or behaviors in development might be slightly different from those that kind of are related to the same traits or behaviors later on in life.
And one of your interests, I know, again, just before we get to the neuroimaging and what you found in this paper, is impulsiveness as a kind of trait. Is that right?
Yeah,
That's
right.
What determines that, and how that might play into likelihood of substance use?
Right. Yeah, and as far as personality factors go, there's a pretty robust literature suggesting that impulsive personality traits of different kinds are very influential on especially the earliest stages of substance use. So early binge drinking behaviors, for example, or initiation of substance use, for example, and that's not surprising because those behaviors also typically begin in adolescence, right, or early adulthood, a time where people are a little more impulsive than they are sort of later in life. And so there's the sort of idea, right, that genetic influences might be shared between impulsivity and substance use, because there's kind of a mediating pathway that some of the genes that influence impulsivity are related to substance use as well.
So, but impulsivity may not be universally bad, right? If it's part of curiosity, and that's what we're explorers and people who also venture into brave new worlds, shall we say. So
absolutely. Yeah. There's sort of a kind of a adaptive feature of risk taking and risk preference, maybe, that can be fairly advantageous if it's not kind of also in combination with a lack of forethought or a lack of self-control.
Right. So getting to the details of your study, can you describe what neuroimaging is, at least what you guys were doing, and then say kind of what were your observations?
Yeah. So in terms of the neuroimaging, particularly in this study, we're talking about MRI scans, which kind of provides a metric of the structures of the brain in different kind of, so like volume of different brain structures or surface area. When we talk about the cortex more specifically, we're often interested in how much surface area there is on the cortex or specific regions of the cortex in general, or thickness of different regions. And all these variables can be kind of calculated based on these MRI scans. And so as part of the ABC.
And MRI, if I could just interrupt for a
Yeah.
second, our listeners probably know that MRI stands for magnetic resonance imaging, and it's where you kind of are shoved into this gigantic tube with huge magnets that allows, in a magical way, to recreate an image of the brain with a lot of detail in terms of the kind of structure, like the cortex, which is the outer lining of the brain, deep structures, and so on and so forth, right?
Right. Yeah. Yeah. And so we took these scans of about 10,000 of these 12,000 kids at baseline, and
And brain images. Wow.
Yeah, 10,000
That's
brain
huge.
images.
How do you keep the kids still in there? Are they okay?
I'm not entirely sure. I do think there are, some of them are not, right? And there are quality kind of data control procedures to account for too much head movement and things like that. But
Sure.
yeah, it's an enormous data set, really. And so based on these scans, you can kind of pull out different pieces of information and use those as variables to test for associations. And so we looked at what are kind of global metrics of the brain, like the entire volume of the brain, the entire volume of the cortex, the entire volume of the subcortical regions, which are kind of inside and underneath the outer cortex. And in all, we looked at 297 different kind of metrics across the entire brain from these baseline scans, right? And then we took these kids who had been followed up at this point based on the current data release, like ABCD releases data every so often. We have data on their substance use from baseline through the third yearly follow-up. And so what we did was we kind of grouped kids into those who at any point during those 3 years had tried some substance, and we later kind of broke it down by substances to some degree, and those who hadn't, right? And then we looked at whether among the kids who had used substances and those who didn't, were there any brain differences that were kind of evident at that baseline scan.
So evident to begin with before
Right.
they ever started.
Yeah, so that was sort of a follow-up. At first, we just kind of looked, I guess you could say, sort of correlationally, but then we did kind of follow up and restrict the sample a little to look at, okay, who are the kids who, some of the kids had come in to the study having already used substances. So we kind of restricted the sample to those who hadn't at baseline used these substances, and kind of followed up our analyses to say, are the same associations kind of coming out, which would, as you said, sort of suggest that these brain differences precede substance use, and the initiation of substance use in this case couldn't have caused these brain differences because they occurred prior to substance use ever happening.
Right. Okay. Tell us kind of what you found.
Yeah. So the kind of the main takeaways were, and some of these are kind of consistent with what we have seen traditionally in neuroimaging studies related to substance use and substance use disorders, and some are sort of the opposite of what we kind of expected to see. So the first sort of set of findings are that for the global metrics that I mentioned, whole brain size, whole cortical surface area, volume, things like that, these were actually larger or greater among those who had initiated substance use.
So they had kind of bigger brains.
They did, yeah. They had bigger brains, essentially, right? Yeah, and typically, previous research has suggested that for substance use disorders, more specifically, right, the kind of the smaller cross-sectional studies I mentioned, we see actually smaller brains, which has been largely kind of interpreted as kind of neurotoxic effects of substance use, leading to some amount of atrophy in the brain. And then we kind of broke it down more regionally. So there's different, the brain can be kind of divided into different regions. And we saw sort of a pattern of a thinner frontal cortex. So frontal cortex is kind of the frontmost part of the brain, above and behind the eyes. And we saw that region, those regions of the brain were generally thinner among those who had initiated substance use, whereas other regions that were associated with substance use were actually thicker apart from this frontal region. So there's kind of an imbalance between potentially the frontal regions in terms of thickness and the other regions.
So just if I can clarify, so
Yeah.
you're looking at basically the front of the brain, and the cortex is where the gray cells are, roughly speaking, right? Because below that is a lot of connecting cable, white
Right.
matter.
White matter,
I mean,
yeah.
just to dumb it down a little bit. And so you're saying in the front part of the brain here, there was a little bit of thinning in people who were destined to use drugs. Is that right?
Yeah, in the kids that
But
went on to
overall,
use.
their brains were bigger because other regions were thicker, et cetera.
Yeah, yeah,
Yeah.
yeah. And and um and then in terms of the subcortical structures, kind of as a group, um, not kind of pulling apart one structure versus another, kind of beneath the cortex, we saw that those were also larger, which again is kind of the opposite of what you might expect to see based on prior research for substance use disorder.
Okay, and that was a general finding. Now, as you know, we're the Center for Medicinal Cannabis Research, so we're also kind of in cannabis specifically. Was there anything you were able to tease out related to cannabis initiation?
Yeah, and sort of in contrast to what I just said in terms of overall the pattern being larger subcortical regions, for cannabis, we actually saw the right caudate was smaller among those who went on to initiate cannabis use. And there was a couple other regions, the left precentral gyrus, which is kind of at the top of the head and sort of separates sort of roughly the front of the brain from kind of the back of the brain. And that was a little bit thinner. And then there was a finding of the in the parietal lobe where the volume was lower.
So we don't always know exactly what each of these structures does because, of course, the brain is pretty interconnected and we need the networks are functioning and so forth. But in general, can you explain what are some of the things that the caudate, first of all, where the hell is it? What is it thought to coordinate? What kinds of behaviors and such?
Yeah. So the caudate is part of the striatum, which itself is sort of part of the basal ganglia. And so this is a fairly deep structure kind of in the middle of the brain. And the brain is typically, when we study it, kind of split down the middle. You've got a right side and a left side, and there's kind of a caudate structure on both sides deep within the brain. And the caudate specifically, and also the basal ganglia more generally, are kind of thought to be involved in reward processing and have kind of connections from those regions to all over the brain, but in particular, the kind of the frontal regions. And so there's kind of thought to be this sort of dynamic top-down, bottom-up sort of action going where there's kind of approach reward-related behaviors in these deeper structures that are kind of then evaluated and, to the extent necessary, inhibited by kind of more frontal cortical structures that kind of are thought to govern decision-making and self-control and kind of just general information processing. And a lot of the research on the caudate and the basal ganglia sort of suggests, along with kind of this reward idea, that these are important structures for the addiction process, that sort of the connection between these deeper structures and these frontal kind of brain structures undergo some amount of dysregulation that kind of perpetuates potentially the addiction cycle.
So again, kind of simplifying it a bit, part of the known sort of functions of the caudate is seeking more reward, wanting to have more fun and stimulation. Would that be right?
Yeah.
Yeah. It's also involved, I think, in kind of planful sort of motor behavior, right?
Right.
Things along those lines.
Yes.
There's also some memory functions, I believe, particularly in the learning area. Is that right?
Yes. Yeah.
Yeah. Well, it's got complicated things. Well, so that's really interesting. So I guess would one hypothesis as a result of this be that these kids who, if I could say, are destined to try cannabis at least, kind of have more more of a drive toward rewarding and interesting experiences, something like that.
That's possibly,
I mean, as a hypothesis.
yeah, I think that's a reasonable hypothesis based both on this study and kind of what we know about the caudate in the literature, that there's some aspect of reward seeking and kind of novelty seeking sort of component to this connection, potentially in line with kind of trying cannabis for the first time.
Okay. Now, of course, your research is ongoing. It doesn't say that cannabis or other drugs can't also do things to the brain.
Right.
Yeah, I mean, they obviously have to do something temporarily in order for some to experience whatever they want to experience.
Yes.
The more important question might be, is there a permanent change in the function or structure of the brain as a result of chronic use of drugs? And this ABCD study is going to be able to, I think, shed light on that as well. So there's the propensity, and then what's the feedback as well.
Absolutely. Yeah. Yeah. And so I think this is another kind of debate that is sort of best settled by saying it's both to some degree, right? That's kind of what our brains do. They influence our behavior, and then different experiences or different substances or anything like that kind of then has an impact on the brain. And whether those are permanent or not, I think is still largely kind of unknown, and the literature is kind of mixed on that at this point. And I think, as you say, the ABCD study will be useful in kind of getting more information about those kind of back and forth.
Yeah. But one thing we do know from the existing literature even is there's a lot of evidence that the brain is really resilient, and that what you may see, for example, in looking at brain images of alcoholics who are wet, if I could call it that, change over time in alcoholics who then don't drink for 6 months or a year or something like that.
Right.
The brain seems to recover significantly structurally and such.
Right. Yep.
So that's another factor that needs to be considered. Are you evaluating people during active phases of substance use versus after they've stopped potentially? Well, great. Anything else you'd like to say in terms of the implications of this work, and assuming the findings continue to be replicated?
Yeah.
What do you think? What's the take home here?
I think, other than the kind of broader take home that we should be thinking about kind of this from a developmental perspective and from a kind of a dynamic perspective, right? There's no kind of one, and, there, as sophisticated as our imaging techniques are, they're probably still likely an oversimplification of the actual underlying biology, right? There's probably something going on that's sort of related to a smaller caudate that then kind of influences things, but the actual, just the size of the caudate itself may not have kind of the mechanistic implications that the results might suggest. But for the study more specifically, I think, in general, the effect sizes we saw were fairly small. So this necessarily deterministic to any degree, and these are not data that can be used clinically at this point. And I think part of the reason we see these small effect sizes is because human behavior is extremely complex, right? And involves lots of different brain regions and different aspects of brain regions that probably go beyond simple measures of size and thickness and things like that. But also the behaviors themselves are very complex. And we're talking about, in this study at least, the very earliest stages of substance use. So many of these kids will not go on to develop a substance use disorder. They'll sort of try cannabis and either never try it again or sort of periodically try it, things like that, which is another reason that following up these kids across this decade will be really important to kind of tease that apart.
Yeah. Yeah. And the other thing is, you mentioned earlier, in some aspects, brain volumes of kids who then later on started substance use were bigger to begin with. So the old thing, bigger is not necessarily better. But is that related to the fact that maybe the process of myelination and kind of, refining the brain's structure may be less complete in those kids? Or again, we're speculating what this means, but what do you think?
No, but I think that it sort of highlights that what we really want to examine, I think, is childhood and adolescence are times of really kind of drastic changes in the brain in terms of its development. And what we really want to know, I think, is, those who go on to use substances or are using substances, are their brain development trajectories kind of different in some way from kids who don't, right? And so these brain scans in the study that we're talking about are at 9 or 10. And so we don't have a good idea of where these differences even still kind of came from. Are they things that kind of were onset early, even in utero or early in life, or kind of what are the mechanisms then that lead to these differences in brains, and when did they occur?
Great. Anything else you wanted to say by way of wrap up? It's been a wonderful talking to you, and a very exciting, set of studies that you're involved in.
Yeah, and so one thing that we didn't explore in this study, as I just sort of mentioned, is kind of now the question in my mind, at least, is where do the brain differences arise from, right? Are they influenced by genetics? Are they influenced by environment? And so that's something that we're actively kind of working on. Are there aspects of the child's kind of early environment, like socioeconomic status or neighborhood deprivation indices or pollution, things like that might be linked to these brain differences? And simultaneously, are there kind of genetic risk factors for substance use disorder or impulsive traits that might be linked to these brains, to sort of examine the extent to which brain differences are actually an intermediate influencer on these behaviors, or kind of mediate these more broad environmental or genetic influences.
Well, and to your point about early influences, they can also be intrauterine influence, right? So before the baby's born even.
Right, yeah.
Well, Alex Miller, thank you so much for this really informative conversation about the chicken and the egg. And the answer is you need both, and we need to talk to each other a little bit.
Yeah, absolutely. I'm not sure we'll ever solve that completely, right? It's both. And thank you for having me. I appreciate the opportunity.
Well, thank you for taking your time, and good luck with your research. And let's have you back when you learn more.
Sure. Sounds great. Thank you.
If you liked this podcast, please subscribe so you don't miss future episodes. For more information on the Center for Medicinal Cannabis Research at UC San Diego, please find us on social media at CMCR_UCSD on Twitter or X, Instagram, and other social media platforms. Or visit our website at cmcr.ucsd.edu. Thanks for listening.